I've had to think about my lab more as I start this new synthetic biology effort and what is it that we work on. And I have sort of divided it into these two large areas, one around, I guess you could call it medicine or health. And there, we've had some interesting projects recently, and this was before COVID. I should add that another driver for me is I have very unusual sources of funding compared to most people. Again, in sort of the more engineering vein, I have money of support, not so much from the NIH, but DOE, defense, other government agencies that you often don't find funding in the medical school. So for example, and as I said, this was way before COVID, we got fascinated by a project that we did together with IARPA, which is the it's the DARPA of the intelligence part of the government of DNI. And you may think they're dark and all that. Actually, they are an amazing group and they do tons of things. And one of the things they were interested in, as I said, way before COVID, as I should say, many agencies have been interested in, is how do we anticipate how bad a particular virus might end up being? Sound important?
Seriously.
So if you have an emerging virus, how do you figure out if it's going to be bad or not? And remember, this is a case of, I'm not a virologist, so there may be people who could answer this question much better, but I decided to deploy the awesome power of synthetic biology, and I guess you could say systems biology, to set up large-scale screens of viral genes to look at how much they impact the innate immune system, which is the first line of defense when a virus is affecting a cell. And it is known that viruses have genes that make proteins that will inhibit this innate immune response. So we reasoned that viruses that had genes that severely impacted this might turn out to be more pathogenic. That's a stretch. But what we did, it was interesting because in the course of this project, COVID happened. So we were one of the first to order all the coronavirus genes and test them in our assay. And guess what? SARS-2 won. We sort of had this real world proof. Lots of other people had that too, but that was sort of satisfying for us. So that was a very intriguing project. We also have a lot of efforts around protein design, especially protein-based therapeutics that are kind of novel, and we have licensed some of that technology over the years. I should add that work, I work closely with my spouse, Jeff Way. I've been fortunate that we've been able to work together all these years, and a lot of the protein engineering work is really his thing. So that's on the medical side. And then we have this sustainability side.